Hello, friends on the internet. This is AP Bio, 39 weeks, day 17. Today we're studying meiosis. Did you get any hairdo? Mm-hmm. From now on, we shall call him Big Haircut. Da-na-na. Okay, meiosis, part 1. Does anyone know what meiosis is? That is the wrong page that I told y'all, by the way. 1, 2, I think we're on chapter 13. There we go. Chapter 13 in your Campbell Biology book, page 252, for those of you following along at home. Okay. Important concepts. DNA is replicated when? S phase. That's part of interphase. Mitosis, the purpose of it is? To make identical daughter cells for growth, division, repair, or growth, replication, and repair, yeah. So you're making more either for your growing or repairing old stuff or replacing things that have worn out. Meiosis, on the other hand, is, what's its purpose? To make sex cells. Good. We are making sex cells and involves 2 divisions. We're going to go through those and see what that looks like. Because those two are very similar, the steps, but there are also some important distinctions, okay? So, first thing on the notes today, we're going to kind of go through the basics and background. We're getting into heredity here, and we're going to be studying how traits are passed specifically from one individual to the next. Like I said, today we're working on this. Tomorrow we've got a lab on meiosis. And then next week, we're going to start Mendelian genetics. There are 5 sections of Mendelian genetics notes. And so I think that's the last part of the notes for genetics unit. Also, for the people in my class at school, you guys have an AP classroom check on molecular genetics that I will, not a classroom check, a progress check that I will be posting soon. And it'll be due for you guys to sort out and take this weekend, okay? Try to get it done by Sunday night. Okay, heredity. Walker, tell us about that. What does it mean?
This refers to the transmission of traits from one generation to the next by inheriting DNA from the parent for asexual reproduction or parents for sexual reproduction.
Okay, so heredity. How are traits passed on? Most DNA is common information. So if you were to look at our genomes, the majority of the information on there would be the same. 99.99% of the individuals in here would share the same DNA. There's that small percentage, though, that's going to be different that helps make us the unique individuals that we are, okay? But we're mostly the same. All right, genetics. What is that, Tay?
This is a study that deals with the transmission of information in the form of DNA. It can range from studying how traits are passed from one generation to the next using Punnett squares or identifying DNA segments, what we call genes, and the proteins and enzymes that make. It is a huge field of science.
All right, genetics. Genetics is basically the study of how those traits are passed on, or you could say the study of heredity. How the traits are passed, studying how those traits are passed from individual to offspring. It's very important in society because it helps us determine how characteristics are passed on. It helps us determine maybe genes that we need to focus on and research, where things that cause diseases. And there's also a lot of ethical questions involved in genetics. Ethics are?
Right and wrong.
Yeah, right and wrong. So what is right, what is wrong? A good example is embryonic stem cell research. Is it okay for us to do research on cells that could become an embryo? So these are questions that we have to ask ourselves. Is it okay to put 2 different cell lines together from 2 different organisms and see what happens just for giggles? So these are ethical issues that we have to consider and think about, okay? Genes, what are they, Katie?
A unit of hereditary information in the form of a DNA sequence of nucleotides found on chromosomes. Most genes code for some type of protein or enzyme.
Okay, so a gene is a segment of DNA that's probably going to code for some type of protein or result in an enzyme being produced. And it is coded from that DNA. And so we talked about transcription and translation quite a bit, so we won't beat that dead horse anymore. And you guys should be familiar with that. Now, genes help to make up part of the genome, okay? So the genome is the entire genetic makeup of an organism. So all the A, T, C's, and G's listed out. Now, in the second part of the notes here, it talks about the N and 2N. Okay. Here's how I'm going to explain that. N is the number, well, you can think N are haploid cells. 2N are diploid or diploid cells. However you want to say it. Di or bi means two. So, in your body, how many chromosomes do you have?
23.
You got 23? You need to get that checked out. 46. So, you got 46 chromosomes. So, we have 23 pairs though, right? You got 23 from mom, you got 23 from dad. Well, you didn't clarify that, Walker. And there's lots of people on the internet getting confused right now. So, you got 23 from mom and you got 23 from dad. 46 total. The N number, I like to think of it as like our 23, we have 23 different chromosomes that code for different things. So all 23 of those are coding for different genes, different traits. We just get 2 copies of it. So we have 23 different genes. That's our N number. When we get 2 copies of it, that means we have 2N. We got 1 copy from mom and 1 copy from dad. Okay? So locus. Locus is short for location, where genes are located. I don't remember the site, but I'm sure you could use the Google out there. There's a really good site that has like all the chromosomes and all the genes that are known on each chromosome, and it tells you like where everything is located, like eye color, hair color, a gene that could cause cancer, that kind of thing. And so all of them listed out. It's pretty cool to go through, although I don't know how useful that'd be for most of us, but it's pretty cool to see that they've located where those genes are. And maybe in the future, when they're trying to fight diseases, that's a good bit of information to have to know where to look for things. All right, there are 2 types of reproduction that can occur by organisms. So on page 254 in your book, we've got, well, really we're not moving much here, so just kind of hang on. 2 types of reproduction are what? Sexual and asexual. So asexual reproduction is used for what purpose? I'm not going to read through all this. Y'all can read through it if you want to, but we've talked about this quite a bit, so I just want to elaborate a little bit.
Really quick reproduction.
Okay, so asexual is for really quick reproduction. Why would you want to replicate really quickly?
Like take over something like that.
Yeah, to colonize an area very quickly. So if you think of bacteria or fungi that are able to sexually and asexually reproduce, you're going to pick asexual reproduction if you have a good environment and you want to take over that environment as quickly as you can. Okay? Now, sexual reproduction, its main purpose is diversity, production of diversity. Now, some individuals, that's their only option, so that's okay. Just like some individuals, they can only asexually reproduce, like yeast. The sexual life cycle has never been observed in those fungi. So for sexual reproduction, the main objective is to achieve diversity. So in individuals, like if we all look around the room here, none of us are the same. If you look at your brothers and sisters, you're not exactly the same as them, even though you share a lot of the same genes and you both came from the same mother and dad, right? So it just goes to show you not everything is being passed on to the offspring, okay? Only part of the stuff. All right, sexual reproduction. As the song says, it takes 2, right? So it takes 2 to make things go right. This process creates variation and is very important in terms of survival. So we're going to kind of hint at what you're going to see in the evolution unit, which is a couple away. When we're talking about sexual reproduction leads to diversity. The more diversity you have, the more likely a population is to have some individuals that survive and pass on their traits. So when you are trying to get some more diversity, if you have the option, sexual reproduction is the key. Risk of sexual reproduction. What is the main drawback?
Takes a long time.
Takes longer. You got to find somebody, got to wine them, dine them, whatever, and it just takes longer. If you're that earthworm out there or the anglerfish or whatever you are, you've got to, have your courtship ritual. You've got to locate them wherever they are in the sea. And so there's more energy and effort that goes into that. You can see our friendly rooster outside. He tricked one of the hens into mating with her earlier. So I don't know what he's got going on. He's got this thing where he goes in the corner by the coop and he puts his head down and he buries his head almost like up under, like in the dirt, like an ostrich, even though ostriches don't really do that. Anyway, and so like he does that and it's like he's hiding or scared and like a hen will come over and she'll be like checking on him and then he'll jump up and just mount her and start mating with her. And so I think he's trying to trick him. I really do. But who knows? It was very disturbing to see it earlier. Anyway, second period can tell you. Okay. Possible different types of life cycles. This is something we need to consider. Hey, before we skip on though, let's look at the bottom of 254. What is a karyotype? So if you look at the picture here, all it is a...
Not just like a picture of all the chromosomes.
It's a picture of all your chromosomes, right, Walker? Ding, ding, ding. Why would you want a picture of all your chromosomes or of someone else's? Where would that application be useful?
I'm just like seeing if there's any mutation, like chromosomes.
Could you see mutations in the genes?
No.
No.
Like, you can see the number of chromosomes.
Yeah, so you can look at chromosomal number and see if there's any like breakages or major deletions or something added to it. Yeah, and you can take the number. When would you want to do this? When do you think people use this? How about that?
There's like bouncing them or something.
Yeah, a lot of times if you have like in utero, if they notice something that looks abnormal and they're like, there may be this chromosomal disorder, then they'll do an amniocentesis, which is where they stick a long needle in your belly into the baby and pull out some cells and they look at its karyotype and it's pretty obvious to tell if they're missing a chromosome, is there an extra chromosome, is there a piece missing here? And so it's a way of determining major abnormalities, okay? All right then. So karyotypes are useful for that. Now, keep flipping on past the naked people on 255, sperm, egg, zygote. I think I got that part right. If you want to know when you were the smallest you've ever been, I was smaller back in college. Too much ice cream. If you'll think back to about whatever your age is and then add about 8 or 9 months, you'll get back to the point where you were one cell. You were a zygote, little baby zygote. Now you're just a Tay. So you're a zygote. You ever think about going back to that name? No is the answer. So, and we all divided from there. Now, what are the different types of life cycles? Haploid majority. Haploid means half. Diploid means 2. So it means these organisms, they appear, their cells are haploid in nature all the time. And so when you think about a fungi, when you're looking at like fungi, like a fungal organism, you're looking at its cells. If they're not the reproductive cells, those cells are haploid in nature, meaning they only have like half the normal chromosomes, okay? And so in order to go to have a sexual life cycle, what do they have to do? Y'all remember? You have to have 2 cells that fuse together. That's called plasmogamy. You know this coming back? And then the nuclei in them fuse together called karyogamy. Any of it coming back?
A little bit.
And then they undergo what to get back to that haploid state? What?
They just divide.
Remember, these are different ways of sexual reproduction. So that we're discussing. Sorry.
How do they go under meiosis?
So they'd have to. So they've come together. Now they're going to have to undergo meiosis to split back apart and reduce that chromosomal number. The difference is, though, the 2 resulting cells are going to be what? Well, they're going to be haploid, but they're going to be genetically different than the 2 cells they started with. Okay, so the whole point of that is for genetic diversity. Okay, more genetic diversity, better off you are. The diploid majority. This is most other animals. It's a lot of any animal you think of where if you look at that organism. Like if you look at someone in here and you look at their cells, what are you going to see? Diploid cells, good. Unless you have some unique X-ray vision where you can see sperm or egg and you need to turn that off. So, I went too far. And the last one is alternation of generation. That's with plants and some algae. I'm not going to get back into that, but that's where like some stages they're haploid, some stages they're diploid, and they go back and forth. All right, any questions on that? Let's look at the last part of these notes. Somatic cells, body cells, they have 46 chromosomes in humans, remember. They are 2N or diploid in nature. All chromosomes, most all chromosomes are called autosomes. So chromosomal number 1 through 22, all of those are called autosomes. 23, pair 23 is called your sex chromosomes, okay? They're referred to as heterologous, which hetero means different. And that's really only true for men, right? Because girls, you just got 2 X's, so you're still homologous in nature there. But the guys, if you've got X and Y, what that means is that X chromosome and that Y chromosome actually contain different genes. So it's really the only chromosomal pair that's like that, where you have different genes on there. The rest of them all are going to have similar genes. They may have different forms of it, but they have similar genes. All right, germ cells is another word for gametes or sex cells or sperm and egg. And fertilization is a term given to
The fusion of the egg and sperm.
sperm and egg coming together to form a
Zygote.
zygote, and that happens in animals and plants and a lot of things, okay? And the zygote goes on to divide and forms a new organism. All right, so that's meiosis part 1. Thanks for sticking with us. Meiosis Part 2. I'm going to run through this one pretty quick too. I know there's a lot of notes today, but for the 3 of you out there watching, you probably need this. So, meiosis, turn to page 262, actually 258. There we go. So, meiosis and mitosis have a lot of similarities. You'll see prophase, metaphase, anaphase, telophase, the whole 9 yards. Typically in prophase, there are many similarities between mitosis, where the nuclear envelope breaks down. In my metaphase, the chromosomes gather at the middle. In anaphase, they separate. You get the idea. Most of that stuff is the same. Now, what are the differences that you need to know? Well, let's get into those. Well, first, let me ask you this. What's the purpose of meiosis again?
Make gametes.
To make sex cells. So in order to make sex cells, we have to reduce what? You got to reduce the chromosomal number down. So for us, that means going to what?
Just gonna go to N.
23. Good. Now, I'm going to mostly reference sperm production today because it's much easier. And as you all know out there on the internet, guys are much simpler. And to be honest, if y'all remember back to the whole egg production thing, you know, there's really only one egg made from that original cell, and you got polar bodies coming off of it. Y'all remember that? Oogenesis? Where like it has to give off an original 23, and then it has to come back after fertilization and give off another 23 sister chromatids. And so eggs are a little bit more complicated. Sperm, not so much. So let's go through. You might want to draw this out for yourselves. So when you start out with, in meiosis 1, how many cells do you have? One. Dang, y'all are good. Got 1 cell. How many chromosomes is it going to have? So we're dealing with humans right now. Different organisms have different amounts.
46.
Oh, we got 46 chromosomes. And what is the N value?
2N.
What? It's 2N. It's diploid. Good. Okay. What do the chromosomes look like in this first cell? What shape? Kind of like an X shape. Good. Okay. It's not really X. It's more of, for those of you out there, it's more of like this, where you have 2 copies of the DNA and they're held together in the middle, but they typically are drawn kind of like an X. Okay. Now, how many cells are these going to split in? This initial one going to split into? 2. You got 1 cell, you got 2 cells. Okay. How many chromosomes are in each one of these cells? 23. So if you look in your book there, what happens in anaphase 1? Somebody read that for me.
Okay. Breakdown of proteins that are responsible for sister chromatid cohesion along the chromatid arms allows homologs to separate.
Okay, so homologous chromosomes separate. That means like, so you've got, let's say chromosome 1. You had one from mom, you had one from dad. And so your homologous pairs, which mean the same, your chromosome 1, one's going to separate and go with one cell, and one's going to separate and go with the other. Okay? So you've reduced it down to, you got 23 chromosomes, and you've got 2 cells. And the N value is 1 N. And the chromosomes look like?
Just like the little strands.
They're still the same. The only thing that separated are the homologous chromosomes. So you basically had like 2 of these guys. Let's say this is chromosome pair 1, one you had from mom, one you had from dad. One will go with one cell and one will go with the other. Does that make sense? So these have not split. All right. Now, how many of these are going to split into?
4.
So you're going to wind up with 4 cells. Okay? So think of it like if this were sperm production, you'd wind up with 4 sperm. Okay? And how many chromosomes do they have? Each one has 23. Okay? And so we got 4 cells. What's the N value?
1.
Okay, so 1 N. And then what do the chromosomes look like? What? They look like this?
They look
Somebody tell me what happens in anaphase 2. Second important thing.
breakdown of proteins holding the sister.
Too much. Read the part at the top. Or just look at them.
Sister chromatids separate.
Sister chromatids separate. These are sister chromatids. One's going to go with one cell, one's going to go with the other. So you're going to wind up, your chromosomes are just linear chromosomes. Okay? One went with one cell, one went with the other. Does that make sense? So the 2 most important things that happen, meiosis 1, during anaphase, your homologous chromosomes separated. So like chromosomal pair 1 separates. Chromosomal pair 2 separates. In meiosis 2, if I can spell, God bless America. Then the thing that happened here is those sister chromatids pulled apart. One copy went with one cell, one copy went with the other. Okay? Any questions? Now, let's see what else is super important that happens here. The next thing is crossing over. So in your books, let's look at that really quickly. So let's go to prophase 1, and you'll see this. Well, I'm going to keep it up here so y'all can, I'm going to draw some more stuff. All right, so prophase 1, sorry for the bad camera work. Prophase 1, what happens, Katie?
Wait, on the notes or in the,
Whatever, read the notes, sure.
variation is created? Is that what
Variation is created. How's it created?
you-
We just said it.
oh, sorry. I wasn't paying attention, sorry.
Obviously, what happens?
Genetic swapping.
Crossing over takes place. What'd you say?
I said genetic swapping.
Genetic swapping. So DNA is swapped. Good. There is a 1 in 70 trillion chance of you having the same sperm or same egg. So our DNA, the way it splits and exchanges DNA, it's pretty much impossible for you to ever encounter another individual that's just like you. Okay? So even if you would have to have about 70 trillion kids in order to have, 2 that turn out the same genetic content, unless you had identical twins, right? Okay, so crossing over, what is it? What happens there? Now, I had an idea for a gang sign one time, and I'll share this with you people on the internet, but this is more like a gang sign for nerds, so like something you might use to get into that Dungeons and Dragons meeting out there. And so when you have 2 like X-looking shaped chromosomes, okay, so and they cross over in prophase 1. Okay, this is our gang sign. What do we call that structure where those chromosomes cross over? It's in your notes.
Is it just
Tetrad. So tetrad is the term where those two chromosomes cross over one another. Okay? So I'll draw it on the board and let's just kind of look and see what happens here. So this is happening in prophase 1, and the whole purpose of crossing over is to do what?
Variation.
Create genetic variation. So this is like, let's say this is chromosome 1, and here's one set you got from mom, and here's one set you got from dad. When these go to separate, they're all going to be a little bit different. Okay? So you had 2 copies that were about the same, and these 2 were the same. But all of them are going to be a little bit different from each other in the end. Okay? Basically, your mom and your dad are exchanging some DNA on your chromosomes. It's effectively what's happening here. Okay? So after crossing over takes place, you wind up with, they separate out, and parts of this chromosome are going to have some DNA that came from the other parent, and parts of this chromosome are going to have DNA that came from the other parent. And so those sister chromatids, when they separate out, they're all going to be a little bit different. Does that make sense? So it's just a way of increasing the genetic variation. So the more genetic variation in life, the what? Increased survival for a population. Good. The more diversity, the better. Very, very good. And that happens when? Prophase 1. Good. I like your enthusiasm. So, if you look at that picture at the beginning on prophase 1 on 258, you can see that structure. The tetrad is where the 4 chromatids are all overlapped. The chiasmata is where they actually make the kind of X shape where the overlapping takes place. And it's not always at the same place. That's actually going to play a role in how genes are linked together and where they cross over. So it goes through and talks about some probabilities. For all you math nerds out there, you'll probably enjoy that, where it's like, why there's so much variation. But basically, there's a lot of different combinations for your DNA to separate out. Now, I will say this, one other thing about creation of variation. The first one says independent assortment of chromosomes. Meaning, if you think of your 46 chromosomes like 23 pairs of shoes. Ladies, some of y'all know what I'm talking about. So, maybe Walker. So, 23 pairs of shoes, and like each one is a different chromosome. Well, if you got all the left shoes from mom and all the right shoes from dad, when you go to make your sperm or egg, are all those ones you got from mom going to go to one and all the ones you got from dad going to go to the other? No. They're going to separate out independently of one another. So chromosome 1, you might have a left foot go here and a right foot go here. And then on chromosome 2, the right foot might go here and the left foot might go here. So you might have, half the chromosomes from dad going to this one, and you might have, the other half going over here. And since we have 23 chromosomes from each parent, it's never going to be what? It's never going to be equally distributed, yeah. So you're either going to have a little bit more from mom or a little bit more from dad that's being, from your mom and dad, that's passed on to your offspring. Does that make sense? So, sometimes people look a lot like one grandparent or another. You ever notice this? Or maybe you see pictures of your grandparents. Theoretically, you could have a lot of genes, like maybe 15 chromosomes from one of your parents that gets passed on to your offspring, and only maybe, just 3 from the other grandparent. And so there's ways, depending on how it separates out, you're going to have a lot of diversity there. The other thing is crossing over. So we talked about your exchanging DNA. That's another source of variation. And the last one is random fertilization. So random fertilization means that about every interaction that people have when they're trying to mate, there's about 200 million sperm from the video that we watched earlier. Now, how many of those sperm fertilize the egg? 1. So just imagine, back in the day, there was only really one egg when you were conceived, but there were 200 million sperm, and that one determined the way you are. And if there was a different one, you'd be a little bit different. Maybe you'd be a little bit taller. Maybe a little bit shorter. I wish I was a baller. Never mind. Okay. Any questions on that one? Meiosis. Tomorrow we're doing a meiosis lab. So if you guys will, you take care.